Hello, today's topic is apoptosis under the chapter Cell Injury. Apoptosis is a type of programmed cell death, genetically programmed cell death in which the cell activates certain enzymes which degrade its own cellular DNA, nuclear DNA as well as cytoplasmic and nuclear proteins. So it is a type of suicide death program. So what happens here is that the plasma membrane of the cell remains intact, but it undergoes certain alterations because of which it becomes easy targets for certain macrophages which come and scavenge the cell. And apart from this, another point to remember is that unlike necrosis, in apoptosis there is no inflammatory reaction. This is because apoptosis, the inflammatory, before the inflammation can occur, the cell and its fragment is rapidly cleared off. That is why in apoptosis you do not get any inflammation. So apoptosis can occur in physiologic as well as pathologic conditions. Examples of physiologic conditions in which apoptosis occur. One is during embryogenesis. During normal development, certain organs undergo involution atrophy. So that is by apoptosis. Then next example is involution of hormone-dependent tissues. For example, endometrium. During the normal menstrual cycle, it undergoes involution. That is by it undergoes shrinkage. That is by apoptosis. And the next example is decline of leukocyte numbers at the end of inflammatory responses. Then elimination of potentially harmful self-reactive lymphocytes which may act against our own self-antigens. So that is also by apoptosis. Then example is turnover of proliferative tissues. Here turnover refers to old cells being replaced by younger cells. For example, occurring in the intestinal epithelium. This is an example of apoptosis occurring in the intestinal epithelium. Here you can see condensed nuclei and many of the nuclei have fallen off. This is a classic example of apoptosis. Pathological conditions in which apoptosis occurs. One is during DNA damage if it occurs. Then if there is accumulation of misfolded proteins as in cystic fibrosis. And apart from that, infections, especially viral infections, all these conditions can show apoptosis. So mechanism. So the main thing is that caspases are the enzymes which are involved in apoptosis. Apoptosis consists of mainly two phases. One is the initiation phase in which many of the caspases become catalytically active. Second is the execution phase in which many other caspases cause cellular degradation of other cellular components. So these are the main two phases, and within the initiation phase there is one, the intrinsic pathway or the mitochondrial pathway which leads to activation of caspase 9, and second is the extrinsic pathway or the death receptor pathway which leads to activation of caspase 8, and in humans it is caspase 10. And apart from this, the executioner caspases are caspase 3 and caspase 6 which function to actually cleave the inhibitors of enzymes such as cytoplasmic DNAs. So that is an overview of the mechanism of apoptosis. Now we will be seeing each in detail. Let's begin with the intrinsic pathway. What happens that in a normal viable cell, in the presence of survival signals, there occurs production of anti-apoptotic proteins. These are anti-apoptotic proteins of the BCL family. This is very important and it includes BCL2, BCLXL and MCL1. These are the anti-apoptotic proteins of the BCL family. So these mainly function to keep the mitochondrial outer membrane impermeable and thus prevent leakage of cytochrome C. Cytochrome C remains inside the mitochondrial compartment and so cell survives. But what happens in the event of a stress? When a stress occurs, certain sensors of the BCL family are activated. These sensors include, this is also very important, BAT, BIM, PUMA, NOXA, also known as BH3 only proteins. So their main function is to activate the pro-apoptotic molecules as well as to antagonize anti-apoptotic such as PCL2. So this is what happens in the event of a stress signal. There occurs activation of the sensors. Then what happens is that pro-apoptotic become active. So when these become active, these dimerize, form dimers, then insert into the mitochondrial membrane, form a channel through which cytochrome C leaks out. And when this leaks out, we'll see what happens. So this is a closer view and this is cytochrome C. So when it leaks out, it goes and binds. So this is cytochrome C. It goes and binds APAF1 in the cytoplasm. When this goes and binds, it forms a hexamer. It forms a wheel-like hexamer which is known as apoptosome. This apoptosome is very important. This has recently been asked as a question. So cytochrome C attaches to APAF1 and this forms a wheel-like hexamer called apoptosome. This apoptosome binds caspase 9 and this leads to activation. This caspase 9 is actually a critical initiator and this leads to activation of the caspases. Whole cascade is activated. So this is what happens when cytochrome C is leaked out. So apart from APAF1 and the apoptosome complex, see, there are other physiological inhibitors of apoptosis. Physiological inhibitors of apoptosis present in the cytoplasm. So other proteins other than cytochrome C, such as SMAC diablo, these other proteins, what happens is that normally these physiological inhibitors of apoptosis, their function is to bind caspases. They bind caspases. Okay, this is the normal function of the physiological inhibitors of apoptosis. But during apoptosis, what happens is that other proteins other than cytochrome C, such as SMAC diablo, these enter the cytoplasm. These are released from the mitochondria into the cytoplasm. These come and bind to the physiological inhibitors of apoptosis. Thus, as a result of this, caspases are released. So caspases are released and these are free to form initiation complex they act along with apoptosome and they release caspases, and thus what happens is that the caspase cascade is initiated, resulting in a successful apoptosis. So this is what mainly happens in the intrinsic pathway of apoptosis. Next is the extrinsic pathway, also called as the death receptor pathway. So here, see, many cells have surface receptors called as death receptors. And many of these death receptors belong to the TNF family. TNF, that is tumor necrosis factor family. So these usually have a cytoplasmic domain that is called the death domain, and the protein is called as FAS, also CD95, and the ligand is called as FAS-L. This ligand is present in T cells which recognize self-antigens. So when FAS binds with FAS-L, what happens is that 3 or more molecules of FAS, they come together and they form a binding site for an adapter protein called FADD, F-A-D-D, that is FAS associated death domain. So FAS associated death domain comes into action. So what this does is, this becomes an attachment site for pro-caspase 8 molecules. So these pro-caspase 8, multiple pro-caspase 8 molecules are brought together and they cleave each other and this results in active caspase 8 formation. Note that in humans it is caspase 10. So caspase 8 or caspase 10 is formed, and these lead to subsequent activation of executioner caspases, eventually resulting in apoptosis, successful apoptosis. So this is what you need to remember about the extrinsic pathway. There is fast ligand, there is fast and fast L, and this leads to cleaving of procaspase 8. This eventually leads to activation of the executioner caspases. So what have we learned so far? There is the intrinsic pathway of apoptosis, or the mitochondrial pathway, which leads to activation of caspase 9. Then there is the extrinsic pathway, which leads to activation of caspase 8 or caspase 10 in humans, and this eventually leads to activation of the executioner caspases, which eventually results in successful apoptosis. So which are the executioner caspases? This is caspase 3 and caspase 6. So what is the function of executioner caspases? These stimulate other enzymes which cause nuclear fragmentation. Examples include DNase enzyme, which degrades chromatin DNA. So the main function of caspases 3 and caspase 6 is to stimulate enzymes that cause nuclear fragmentation. So that is what eventually happens by executioner caspases. So after chromosomal nuclear DNA degradation, let's now see how these dead cells are removed. So as I already said that apoptotic bodies and apoptotic cells, the plasma membrane remains intact, but the membrane undergoes such alterations that it becomes easy targets, tasty targets for macrophages and other phagocytes. So here mainly it sends out eat me signals. The apoptotic fragments send out eat me signals. So what are these signals? One is phosphatidylserine serine. See, this phosphatidylserine serine is normally situated in the inner leaflet of the plasma membrane. But in case of apoptosis, this flips out. It flips out and then it is recognized by macrophages and other phagocytes. Another important point to remember here is annexin 5. This annexin 5 is used as a marker, especially in flow cytometry, to detect apoptotic cells because it binds to phosphatidylserine. One is phosphatidylserine. Apart from that, these apoptotic cells are also coated by thrombospondin or other natural antibodies of the complement system. These are also recognized by macrophages and other phagocytes. So that is about apoptosis. Next short topic we'll be doing is necroptosis. So necroptosis is a process. It is also a genetically regulated program, but it morphologically resembles necrosis. So mechanism wise it is because it is genetically programmed, it resembles apoptosis. But the main difference from apoptosis that caspases are not involved. So this can be asked, which is the type of genetically programmed cell death in which caspases are not involved? The answer is necroptosis. So necroptosis, the mechanism, it begins just like apoptosis. Death receptors are involved and it forms an RIP1 complex with RIP1 and RIP3. So at this stage, if activation of caspase 8 occurs, then it leads to apoptosis. But in case there is failure to activate caspase 8, then there occurs necrosome formation. This leads to various metabolic alterations, leading to decrease in ATP and increased reactive oxygen species. And this leads to many effects such as lysosomal membrane permeability is lost, all the enzymes leak out and lead to degradation of the cellular macromolecules, and eventually it degrades the membrane also. So cell death, when it finally occurs, it morphologically resembles necrosis. So that is what happens in necroptosis. So necroptosis can commonly occur in case of formation of the mammalian bone growth plate physiologically. Pathological conditions also necroptosis can occur, such as neurodegenerative diseases, Parkinson's disease, viral infections, reperfusion injury are all examples of necroptosis. So that was necroptosis. Next is pyroptosis. Pyroptosis too is a type of programmed cell death. But here caspase is involved. Caspase 1 is involved. So the name pyroptosis is because it is apoptosis with pyrexia, with fever. So here fever-inducing cytokine, interleukin 1 is involved. So here what happens is when the microbial products, they enter the cytoplasm. So these are sensed by a multi-protein complex called the inflammasome. This, when it is sensed, this leads to activation of caspase 1, which leads to interleukin 1 activation. Interleukin 1 is converted from its inactive form to its active form. So that is it about pyroptosis. Next topic is autophagy. So autophagy is actually a survival mechanism under stressful conditions. Here what happens is that the cell eats its own DNA or own contents, and here what happens is there occurs delivery of cytoplasmic materials to the lysosomes for degradation. So it mainly occurs in atrophic cells and in disease conditions such as cancer and in neurodegenerative disorders such as Alzheimer's, Huntington, and also in infectious diseases such as mycobacterial infection, HSV1 infection, Shigella infection, and in inflammatory bowel disease also autophagy can occur. So this is what the main mechanism that occurs. See, there occurs initiation, that is formation of an isolation membrane. Then there occurs elongation, that is elongation of the vesicle. The next step which occurs is maturation of the autophagosome, followed by fusion with the lysosome. And following fusion with the lysosome, there occurs degradation and recycling of the metabolites for further use. So that is it about autophagy and apoptosis, necroptosis and pyroptosis. So now we'll be dealing with a few questions. True statement about apoptosis. Injury due to hypoxia. That is false because injury does not occur due to hypoxia in apoptosis. Injury is mainly due to lack of survival growth factors or due to DNA damage or misfolded proteins. So that is false. Inflammatory reaction is absent and cell membrane is usually intact. So the true statement is Councilman body is associated with apoptosis. This Councilman body actually occurs in yellow fever and certain other conditions. Caspases are associated with which of the following? So we just now read that embryogenesis is a condition. Embryogenesis means normal involution during the development in which physiologic apoptosis can occur. So that is the answer. Light microscopic characteristic feature of apoptosis. The most characteristic light microscopic feature is said to be condensation of the nucleus. CD95 is a marker of. So CD95 is actually another name for FAS protein. So this occurs classically in the extrinsic pathway of apoptosis, that is the death receptor pathway. And in apoptosis, APAF1 is activated by release of which of the following substances from the mitochondria? So this, just remember the diagram, it is cytochrome C, which when released into the cytosol binds to APAF1, forms the apoptosome, which initiates the caspase cascade. Thank you.